Today we have Sven Dorkenwald, who joined the Allen Institute at the University of Washington in September of last year. He received his undergrad degree in physics and a master's degree in computer engineering at the University of Heidelberg in Germany. Sven also received a PhD in computer science and neuroscience from Princeton University, where he developed approaches for reconstruction and analysis of neuronal circuits from electron microscopy images. He spearheaded the Flywire effort that produced a synapse resolution connectome of an adult Drosophila brain. Sven also created a self-supervised approach for efficient annotation of cell reconstructions as a student researcher at Google Research. Here in Seattle, that is, as a Shanahan Fellow at the Allen Institute, Sven is pursuing the integration of neuroscience data sets from multiple modalities. Welcome, Sven. Looking forward to your talk today. Thank you.
Thank you for the very kind introduction. Can you all hear me? Also back in the room? Great. Okay, yeah, this work is connectomics work in the fruit fly, done in Princeton, and it's really a project that is at the intersection of computer science and neuroscience and shows how computer science can really drive neuroscience forward and further that research. As the title suggests, this is all about fruit flies, and I'm sure you all have encountered fruit flies, but you may not be so fond of them just yet. I sure wasn't when I started this project, so I hope that you might be after this talk. Because fruit flies are really juggernauts when it comes to molecular and neuroscientific research. 6 Nobel Prizes have been given to research in Drosophila. A lot of the early genetics research has been done in Drosophila, and the circadian clock has also been discovered there. The main feature, though, of the fruit fly that is important for this talk is the size of its brain, especially when we compare it to that of other model organisms. And as you will see, the size of about 140,000 neurons is just small enough now that we can reconstruct circuits that span the entire fruit fly's brain. And despite its small size, fruit flies still show a wide array of complex behaviors. From grooming to courtship, the males follow the female, they sing with their flies, they pulse them in certain frequencies, and that has all been really well described. They have sophisticated senses that can taste with many parts of their bodies, including their feet. And of course, they can taste, hear, smell, and see with their large eyes. The question really is, though, how these senses give rise to these complex behaviors. And that is really something that has been missing so far. And what would be really useful to analyze that is a map of the brain. So in this map, every node is a neuron, and the connections between them as edges. And such a map of all the neurons of the fruit fly's brain will allow us to trace circuits that start from sensory input down to its output. You might be aware of one organism for which such a connectome has been created. That is C. elegans. It's a nematode, has 302 neurons and a couple of thousand synapses between them. And we can show such a connectome as a matrix where every neuron is a row and a column in this matrix, and every dot here indicates that there are connections, synapses between them. In this case, 2 different types of synapses. We wanted to do that now for the 3 magnitudes larger brain of the Drosophila. To create such a map, we have to image the brain at a very high resolution. Electron microscopy is right now the only method to image a brain at a high enough resolution that we can zoom in and appreciate the neurons at a nanometer resolution. So in this image here on the right, every outline here is a membrane, and within it is 1 neuron. We can identify different features here. For instance, this piece here is called a T-bar. It indicates that there's a synapse. These little dots here are vesicles that contain neurotransmitters. And this here is essentially a synapse where this one neuron here talks to all those postsynaptic neurons here that are indicated with that red star. And by scaling this method up to the whole brain, this was the first data set, electron microscopy data set of a whole fruit fly brain, and that's also what we're going to use in this presentation. Because this data is acquired in 3D, we can flip through these slices and follow these neurons as they traverse the brain. So if you just follow one of these, you can see how it goes away. And if we are successful in following it, we can reconstruct it as it spans the brain. Here I'm showing you 3 of those. And then also by finding the synapses, of which I showed you just one before, we can find all those synapses that are attached to it, and then can find the partners like these 2 here. These are 2 partners of that one neuron. Such maps have been generated for the fruit fly, but they all were partial. So this here is one example of such a map, the largest one that was available before, 25,000 neurons within this part of the brain. Before that, we had other that were even smaller in the visual circuits, and one that just came out last year that contains the entire ventral nerve cord that is basically the spinal cord of the fruit fly. So because they are partial, they only contain the neurons as they're within their volumes, and connections that go out of them cannot be analyzed. But what they all have in common is that they start from these electron microscopy images, and they have to reconstruct the neurons that are within them. So that is basically a machine learning problem, which just comes down to assigning the same ID to all the voxels that are within one neuron. And that is quite similar to other classical machine learning problems that you might be aware of for object detection and segmentation. And it's really that that the field of connectomics has been at the forefront of that, adopting convolutional neural networks early on to solve this task. So we use state-of-the-art segmentation methods that were developed in the Sang lab to apply this to the entire EM data set. So basically every voxel 3D pixel in this data set is annotated with an ID, and that ID refers to a segment, ideally a whole neuron. The challenge, though, is that even though these methods have improved dramatically over time, segmentation in these data sets is really unforgiving. And I want to show you what I mean by that. This here is a segment as it came out of the automated segmentation. And you may not appreciate it right away, but this contains quite a good number of errors. For one, we can see that there are 3 cell bodies in this. One neuron should only have 1 cell body. And it's really only a very small number of errors that were being made, but because the data set, even though the performance is so high, because the data set is so large, there's so many opportunities to make errors. Here's, for instance, one error where this one neuron that passes over is falsely attached to this big one down here. And if I show you how this looks like after this was corrected, this one error here basically was what connected these 2 big segments together. So the problem really is that if your data set is large enough, basically no segment that comes out of one of these automated reconstruction tools is correct. So we need tools that can correct these errors and allow us to correct them through proofreading. And when we started this project back in 2019, really no tool existed that allowed a number of people to collaborate on proofreading those segments. And really, it is a large number of people because while correcting each individual segment only takes a little bit of time, there are so many of them that no individual person could do it. So we needed tools that would allow a community of people to proofread these segments. So what is proofreading? When we zoom in on the segmentation, we can break it up into individual pieces. And that is what's happening during the segmentation process. The first step of segmentation is to create what we call an over-segmentation. Supervoxels, a group of voxels that we believe definitely belong together. And they're basically atomic pieces, and they can be combined through a graph where touching supervoxels have an edge between them. But there's a step, the second step in segmentation is to find this graph, to combine those supervoxels, and then connect the components in this graph are the segments. Now, that allows us to reduce the proofreading problem essentially to a graph problem. And that is where a merge would just introduce new edges into this graph, and the split would remove edges from this graph. And we were not the first ones to do that, but the problem with this is the scale of the data set. Here, I'm showing you a 1D version of this, essentially, a naive implementation. Every dot here would be a supervoxel, and these here are one connected component because they're all connected, and they're all pointing to the same neuron ID. The graph that we're looking at had 2.4 billion supervoxels and about 10 times more edges. We are also now working on graphs that have trillions of edges in them. And every segment in it has tens of thousands or millions of supervoxels. So that makes computations on that graph quite challenging. For instance, just reading all the edges that belong to one segment can take a long time when using Bigtable, which is one of the most efficient, the fastest databases available for this. Even just reading the edges for one segment can take up to 10 seconds. So we had to improve over this. And the insight really was to combine the spatial nature of the data set with the graph. Every edit is essentially just a very local affair. So if we introduce spatial awareness into the graph, we can improve this performance. So what we did is we introduced an octree, which essentially just partitions the entire space into small chunks. And then every level in this octree contains a larger and larger and larger part of the data set. What does that mean for a graph? So here, this is the combined version of the graph and the octree. Here we have a local connected component, and that is combined with this one node here to one connected component that is contained in this spatially underlying graph, and we can combine these components in the octree to larger and larger components. You can think of this as caching the connected component information into these nodes. So what does that mean for an edit? When an edit comes along and we just remove an edge here, we only have to compute the connected components within this small chunk and then create two new components here. And then we only have to propagate this up the tree instead of through the entire data set. And this is what this looks like. So we have two new components, and we never had to touch all the other parts of this graph. This improvement allowed us to reduce proofreading times per edit to about a second for merges and splits, and made proofreading of these data sets possible. We also have implicit versioning, where we can assign timestamps to these edges, and that allows us to retrieve earlier versions of this graph by just filtering this hierarchy with the timestamp. So this is what an edit looks like when a user makes it in our front end. So here I have a segment, and I'm going back and forth here on the left, identifying that there was a continuation that was missing. I'm zooming in, clicking on both sides for a merge that is sent to the server, and that returns it back and merge it back together. Anybody else who is coming across the same neuron now will see this new state of the segmentation. So this allowed us to basically pass on proofreading of this data set to a community of scientists that were interested in analyzing this data set. And we really made it open. Anybody could join this community. We called it Flywire. And the idea was that every scientist wanted to proofread their neurons of interest. That would not take too much time for them. They could analyze the circuit, but together they would contribute to the proofreading of this whole fly brain. And the principles that we put in place were really like that if you want to use neurons that others have proofread, you had to ask them for you had to ask them before you could use it for your own publications. Generally, it was such that people gave each other permission to use their neurons, and because everybody was interested in different questions in the end. So together, we started in 2019, and I am giving you a glimpse here. So every dot here is an edit that was made through Flywire's history. This is up to June last year, where we have made 2.7 million edits together. We're now at about 3 million. And this graph here shows the number of edits over time. And at first, it was really just community members that joined and proofread their neurons of interest. And then in 2021, we realized that we had the opportunity to finish this data set and completely proofread everything that the community has not been proofreading yet. So we put in centralized proofreading, that is basically paid proofreaders that proofread everything else that wasn't proofread before. And but at the same time, we still had people joining. So this graph here shows the number of individual users making at least one edit every week. So we had up to 50 people making at least one edit every week, not just analyzing the data set, but actually editing it. And overall, we had over 200 people making at least 100 edits in this data set. And by the time that we finished, there were already tens of papers written using this data set because it was open and publicly available. This is all the people that are part of the Flyby consortium and contributed to this resource. And in the end, we proofread 139,000 neurons, and it's a graph of 54 million synapses between them. This also includes outputs from different methods, like the synapses. They were published independently. There was another paper that just typed those synapses into those that are putatively excitatory or inhibitory, meaning they excite the postsynaptic side or they inhibit it, preventing it from being excited itself. So it's really a collaborative effort by many people together that created this resource. And so over the next slides, I want to show you some highlights what you can do with such a data set in hand. The first thing really is we have to categorize those neurons. And that was a collaboration with folks in Cambridge, UK, that is. And we first typed every neuron as being intrinsic to the brain, meaning no outside connections whatsoever, those that are completely contained in the optic lobes. So basically the visual processing centers of the fly that are sitting on the side, those that are in the central portion of the brain, and those that go back and forth between them. Then we have those neurons that are projecting out of the brain. Here, the biggest portion are those that are projecting into the ventral nerve cord. Remember that is basically the spinal cord of the fruit fly, and those that project into the brain, up from the spinal cord or delivering sensory information. A lot of this, as you can see, is vision. So from the side, the fly is a very visual animal, but there's also gustation, olfaction, and all those senses are included here as well. So the next thing we wanted to do is create a connectivity map, just as the one that I showed you at the beginning from C. elegans. The problem, though, was that if you have 139,000 neurons, if you square that, every connection is smaller than a pixel on your screen. So we had to summarize them by brain region. The fly has brain regions that are called neuropils, and they subdivide the brain into about 78 of them. And we can now create a map of connections between them. So this is a map of the number of neurons that project between the individual neuropils. And just to highlight this, if you look at this row here, this would be all the neurons that project out of this one neuropil here. And one of those dots is a whole group of neurons that project between those two neuropils. When you glimpse at this, the first thing that becomes apparent is that a lot of the weight of this matrix is in the upper left and the lower right quadrants. And that means that a lot of the connections, they stay in one hemisphere. And we quantified that also on a per neuron level. And this is just the neurons in the central brain. We basically quantified how many of the synapses are in the other hemisphere, in the contralateral hemisphere. And most of those have all their synapses on the same side. But there are some that are only projecting to the other side and some that are in between. So the majority really stays on the one side. I find this group here very interesting because it means that there is a direct left-right connection that has no same-side connection. And that is actually something that also others have found also in the larval stage. So that's something that might be contained through development. So for the second thing, I want to zoom in on one specific circuit. One thing that I was not aware of when I started this is that the fly has more than 2 eyes. And if you look very closely on the top of the brain, here, you can see 3 simple eyes. And those are called the ocelli. They have, they're very small. They only have 80 to 100 photoreceptors in them, and the lens is very under focused. So it's a very blurry image. You will not do any object detection with those, but they are sensitive to UV light. So they are thought to be important for stable flight. Though they hadn't been really researched much before. And so we look deeper into them. The first thing is that we don't have the eyes in our data set, but we have the photoreceptors. So we were able to reconstruct those. That's just like the last piece of the axon that's in our data set. And they innervate, we know which eye they come from the right, median, and the left. And they innervate one small region in the data set that can actually be subdivided into medial, left, and right. So we know exactly which neurons downstream receive input from which eye. And the main neurons that receive input from them are called the ocellular projection neurons, and they are huge. This is a view of those neurons from the back of the brain. So you're looking from behind the fly onto its back, and there are 14 of those, 12, and then 2 that are actually descending neurons. So they are going directly to the ventral nerve cord onto motor neurons. That's a very short circuit. And the other 12, because we have these tapped synapses, we know that 6 of them are inhibitory and 6 of them are excitatory. When we take a closer look at them and we see from which eye they receive input, it becomes clear that each of those only receives input from one eye. So there's some retinopathy involved. So you can assign basically an eye to each of these projection neurons. And if you look very closely at them, you can see that these blue ones here, they cross over, whereas the green ones, they stay on the same side. So what's basically going on here is that we have a circuit where the inhibitory neurons, they project on the same side, the excitatory neurons, they project to the other side. And they target the same downstream neurons here with a lot of synapses, hundreds of synapses on the same target neurons. So this is a circuit where we have ipsilateral inhibition and contralateral excitation. And that is reminiscent of what people have proposed in the 80s of a Breitenberg vehicle. In this Breitenberg vehicle, it's a very simple automated vehicle. It has basically 2 sensors and a connection from the sensors to the wheels. And when we put here a source into it, the sensor that is closer to the source is driven more. So in this case here, because we hook up the other side positively, this wheel will spin more because it's closer, because it's connected to the sensor, which is closer to the source. What this does is it will orient our vehicle towards the source. So that's called phototaxis. And essentially, this circuit here mimics that. It also mimics it for the inhibitory connections that inhibit the same side wheel, essentially. So we have basically proposed a circuit that has a proposed, a hypothesized function of phototaxis. And that, you can imagine, is very useful if you want to fly stable. Your eyes are sensitive to UV light that wants to orient you towards the sky and keep you basically upright. This is just showing you how you can look at the circuit and come up with an hypothesis that you can now go and test on other flies. We, of course, can't do all the analyses that are out there that one could do with this data set. So the most important thing is that it is publicly available. We developed a tool called Codex, where you could now go, and anybody else can go, and download the data and analyze it. And many have done so. One thing I want to highlight is the first analyses that have tried simulations of this data set. So this work here, they basically activated some sensory neurons that are known to be responsive to sugar and inferred downstream activity of neurons and replicated this in living flies. And they showed that the activity that they inferred matched roughly what they measured in new flies. I'm putting simulations in quotes because there's a lot of questions left how we actually simulate those neurons. We know the number of synapses, but what does that actually mean from an activity standpoint? So there's a lot of work left to do. But yeah, I wanted to highlight that. So in the last two slides, I want to go a little bit beyond the fly and fly connectomics, because we have the field have analyzed more than just the fly. Every dot here is a EM data set that was acquired. On the y-axis is the size of this data set. The fly data set is located here. It's one of the larger ones, but by far not the largest. This is a log scale. And you can really see that the field has increased data set volumes exponentially over the last decade. And the goal here really is, for many of us, to scale towards a whole mouse brain, which is probably, depending on who you ask, 10 to 20 years away. But the first data sets, oh yeah, and this all is of course only possible because we have automated machine learning tools to actually analyze these data. Nobody's gonna do this manually. And the largest data sets today, they are about a cubic millimeter in size. That's about a petabyte in raw EM data. We have one from human cortex, one from mouse cortex. And those really pose new problems that go beyond what we had to address in the fly. Just to highlight this, these are two neurons from the mouse cortex data set. These are huge neurons. They connect over hundreds of microns. Just one synapse up here that you have to find between those two neurons. The challenge really is that proofreading those right now takes about 50 hours per neuron. So there's no fully proofread data set out there at the scales, and we have to address these challenges to analyze these data sets, or even 10 times, 100 times, 500 times larger data sets, which would be a whole mouse brain. So a lot of challenges left, but the fly we have now, though there will be more fly data sets coming. I want to stop here, and I want to thank all the people that were involved in this huge project, the entire Flyby consortium. Most of the work was spearheaded at Princeton, but strong collaborations to the Allen Institute and the LMB in Cambridge, a lot of funding from the NIH. Yeah, thank you.
Thank you, Sven. That was great. Questions for Sven? I'm curious about the proofreading. Like when it's like it kind of caught momentum, was that because of more researchers joined, or was some citizen science involved in this too?
Yeah, we had actually also some citizen science involved, but it is in some ways a self-propelling thing, because the more proofreading you have, the more of the graph you build, the more of your partners already proofread, the more valuable adding one neuron to the graph becomes. So there is certainly a self-propelling factor that is involved there. And I think for many, it was, I mean, you do your lab experiments to do one analysis, but here you can also go and you invest some hours in proofreading, and then you can do an analysis too. What certainly also contributed was that we started in 2019. In 2020, everybody went home. You had something you can do from home and analyze, so that's definitely also contributed a bit there.
And how do you work? Did you have a visualization platform or like interactive? How did?
Yeah. So I briefly showed this earlier. So there is and I mean maybe I should. I'm doing what one should maybe not do in the talk, but I can show you. You can go here and look at the neurons in real life. So this is the neuroglancer, which we adopted for the Fly project. It has a 2D view here on the left, a 3D view on the right. You can click on any neurons, and they will show up on the right, depending on how good the internet is. And yeah, so this is how you can load those neurons and interact with them. And proofreading is done through the same platform, though you have to have, of course, the permissions to do that. Not everybody can. Yeah. Yeah.
Scans from just one fortunate drive.
These scans are all from one fly. It's a very intricate. I mean, there are people that work only on acquiring these data sets. You have to cut at 40 nanometer thickness. You have to collect them on, in this case here, this data was collected on a film that has tiny holes in it. So that's, people are just building automated systems just to collect the data, and then you can feed it through a microscope. Yeah, so this is one fly in the end. Yeah, this one does not. The mouse data sets, they were renamed them after cartoon mice, like Pinky and Minnie, but this one doesn't have a name. Yeah, it's like
So, you have timestamps for when you created the neuron set with the algorithm and also the end of the proofread, right?
Yeah.
So why not do like an automated system of like training a algorithm to, or like, okay, this is a mistake that this made, and also like this is the final edit. Is that something you considered?
Yeah, 100%. So I think this is the next step for us to automate. The segmentation, you can imagine a segmentation algorithm that started it, that had a field of view that is basically like so much. That's what went into this, and it had to make a decision here. But we humans, we look at those neurons as a whole, right? So now we have these proofreading edits, and we can train new machine learning methods that can take this whole segment, reason about it, and say there's an error here and there, and we can use that data to train that. But yeah, these are projects that are open and up for grabs. We have millions of edits, not just in this data set, also another data set. Yeah.
Variation in fly lengths? Like, what if this fly has some kind of mutation or weird anatomy?
Yeah, that's a very important point. The flies are more stereotypical than mammalian brains are, and but there's still variation. And at the beginning, I showed that there are some data sets that have been acquired and this one up here. So our collaborators in Cambridge, they compared ours and theirs from two different flies where they overlap, and they tried to get at this question a little bit: how much variation is there? Two flies will not cut it like that. You will need many more to make those conclusions. They found a good amount of stereotypy, like agreement between the two. You of course don't know if the differences are technical or biological. That's another source that you have to figure out. Yeah, and so, but they also found interesting differences between the two. And you don't know if that is due to how the flies were kept. One was maybe fed more than the other, and that kind of leads to it being more sensitive to certain things. Yeah. I think that the meta point here is that our field has really been struggling and working hard to even scale to this, so that an N of 1 so far was the only thing that we could do. But I think down the road, the goal is to do multiple brains, compare them. You have mutations in some of them. You have maybe a certain diseases even that you can compare and see what they actually do in the brain. So that's, I think, it's once you understood what is biological variation, you can go and try to answer these questions.
You touched on this a little, but what progress has there been made on other animal models and their connectomes, so like mouse or even macaque?
Yeah. So what I showed you here at the end, that these were the largest. Not do this. These were the largest data sets that exist. So there has been proofreading in those, and we have. So this year is also collaboration between Princeton and the Allen Institute. We have on the order of 1,000 neurons proofread in those that are publicly available, and anybody who wants to analyze them can analyze them. So there are mammalian circuits that are out there. What is really nice about the fly is that because you have the whole brain, for most of those neurons, you know all the inputs, all the outputs, and that gives you a level of analysis that you can do that you cannot do, even if you have this data set fully proofread in the mouse. Though there are already amazing papers that use those data sets too, and a lot can be cleaned from that. Yeah.
mentioned something about proofreading, right? Is there a mechanism in place for consensus thinking? Like, what if someone proofreads it wrong? Like,
Yeah. Yeah. So we basically took the approach of Wikipedia style. Like, somebody else can come and correct something that somebody else did. But we had a system where people could mark a neuron as proofread, and then others could see that, but they were still able to make changes to it. Now, of course, somebody could go in and mess something up now after all, so that is still possible, but in our experience, that does not happen, or very rarely. It's the chance of fixing something, because there's always an error left to fix, outweighs the chance that somebody might introduce an error.